Chapter 1, The Study of Life. This chapter is going to be a little bit different than some of the other chapters in the book. This one talks about evolution and biology in great big broad brushstrokes. In this chapter, we'll be covering the fundamentals of life, and we'll be tying these fundamentals of life to three theories that are very important in biology: the cell theory, the theory of information, and the theory of evolution. And then lastly, we'll talk about the business of doing biology. What are the 5 fundamental characteristics of life? The first 3 are cells, replication, and information. That is to say that all organisms are made of at least one cell. All organisms have to replicate, and to replicate they need special information, and that information is found on genes. The other 2 characteristics of life are energy and evolution. All life needs energy. I often think of Star Wars, like may the force be with you. So that force is energy. And then evolution is life is dynamic. Life is always changing in response to an ever-changing environment. Before we can get started, we need to talk about what is a theory. A theory is an idea that explains very basic general phenomena, and the important thing is that it's supported by a wide body of evidence by different scientists over different places, and this information has accumulated. So we don't use it like in everyday language where we think it's speculation or a guess. This is something way more concrete. So the theories that we're going to talk about are the cell theory, the theory of evolution by natural selection, and the chromosome theory of inheritance. And this gives us the what, where, and how about life. To talk about cell theory, we need to start talking about Robert Hooke. In 1665, he devised the first microscope that magnified cells 30 times. And when he looked at these cells or compartments, he called them cells. Then later on, von Leeuwenhoek magnetized it even 10 times more. So 10 times 30 is 300. And the organisms that he saw, he called animalcules. By the 1800s, many biologists, especially from Germany, were claiming that all life was made of cells. Here are some examples of living and some dead tissue. So on the left you have cork, and then on the right you'd have something that you would find in a pond. In the 1660s, Robert Hooke and Anton von Leeuwenhoek were the first to observe cells. But the question is what do we mean by a cell? A cell is a highly organized compartment, and whatever you see in that cell is very different from the outside environment. So the cell theory means that all organisms are made of cells, and all life came from pre-existing cells. Now, there's a classic experiment that talks about how we know that this occurs, but before we can talk about it, we need to go over what is theory, hypothesis, experiment, and prediction. So we already know that a theory explains some very basic patterns. A hypothesis is an educated guess, but it's really something more. It's something that can be testable and be proven falsifiable. And then we need to establish an experiment. An experiment is where we have treatments in a controlled environment and we collect data. And this is made from prediction. So we take these observations and we use them to see whether our predictions are correct or not. So this experiment by Louis Pasteur actually looked at the cell theory that says that all life comes from pre-existing cells versus spontaneous generation, that things just automatically appear. Something like maybe that you would see in Harry Potter. But the truth is we know now that all cells do come from pre-existing cells. So what was the classic experiment that actually proved that this had occurred? In Louis Pasteur's experiments, he hypothesized that all cells arose from pre-existing cells, and cells do not spontaneously generate. And to do this, his experiment consisted of 2 glass flasks, and both of them contained sterilized nutrient broth. So what was the treatment? One flask, the neck was short and stubby and facing upward, and in the second flask, the neck was a swan neck. It was open to the air, but after a curve actually in the neck. Here you can see how the experiment was laid out. We see that it's a controlled experiment. We see that there's 2 glass flasks on both sides. They both have nutrient broth. And the only difference between the two in this experiment is the shape of the neck and how it's facing up into the atmosphere. Notice the S, or the swan neck. On the right-hand side, you can see that there's a water trap. And even though it's facing up the opening to the air, actually the water trap prevents movement of air down into the broth. So as you can expect, we will see growth happening in that neck that is open to the air, and in the S, or swan neck, we will see no growth in the broth. Cells must replicate for life to exist, and that's what Louis Pasteur's experiment showed. All cells present in multicellular organisms have descended from pre-existing cells, and they all are connected by common lineage. And evidence also shows that there was a chemical evolution before there was a biological evolution. The next theory is the chromosome theory of inheritance, and this was proposed by Sutton and Boveri. It says that heredity, or genetic information, is encoded on genes, and these genes are located on chromosomes. And chromosomes were discovered in the mid-1950s, and so we know that chromosomes are made of DNA, and on DNA we have genes. These are regions that code for specific traits. So on the double helix, we have 4 building blocks: A, T, C, and G: adenine, thymine, cytosine, and guanine. And a combination of these actually will code for a particular protein. And we know that most life is made of proteins. So this sequence is something unique and will code for a specific protein that affects an organism's growth and reproduction. Here, Watson and Crick were the first one to propose what a DNA molecule looked like, and it looks like what you see right in front of you, the double helix, where the red ribbons represent sugar and phosphate groups, and then you see that they're connected by complementary base pairing of A, T, C, and G. A does always pair with T, and C pairs with G, and this pairing is how we know that DNA is copied, and it also preserves the information that's coded on DNA. And again, we see the double helix, and we see the complementary base pairing. Another important thing about DNA is this idea of central dogma, and this is the flow of information from DNA to protein. So the sequence always goes DNA, RNA, and protein in most instances. DNA, RNA, and protein. In this drawing in figure 1.4, you can see that we're looking at a eukaryotic cell where the DNA is found inside the nuclear envelope. A section of DNA has been copied by messenger RNA indicated by the yellow ribbon, and this yellow ribbon will exit the nucleus and move to the cytoplasm to find a ribosome. And at the ribosome, that information will be translated into a protein. And here you see that that translation has encoded for a salamander. RNA and proteins. RNA is a molecule that carries the information from DNA to the ribosomes to actually synthesize proteins. Proteins are not only used for structure, but they're also used to guide metabolism. DNA is copied to pass genetic information from cell to cell or from one organism to its offspring. And in most cases, DNA is highly accurate. In some cases, there might be a mistake, and that is often a permanent change in the DNA. And this leads to more variation, and it's commonly called a mutation. So DNA sequences do change, and these can lead to heritable variations, and this is what underlies the diversity of life itself. Life also needs chemical reactions to create energy. So this flow of information requires energy, and energy in the cell is typically in the form of ATP, adenosine triphosphate. And adenosine triphosphate can be used to help build DNA and to build proteins. The source of energy ultimately comes from the sunlight, and specific organisms such as plants and bacteria can convert light energy from the sun to chemical energy. And this chemical energy is in the form of sugar, and this sugar helps to generate ATP at the cellular level. In this figure here, 1.5, you see an animal like the butterfly dependent on the plant for energy. Now we're ready for the third theory, and this is the theory of evolution. Darwin and Wallace are considered the fathers of evolution. And evolution states that species are related by common ancestry. So if we look at the cell theory, if all cells come from pre-existing cells, one cell comes from another cell, and that cell comes from another, and so on and so forth, then we see that we are all related to one another. Another way of saying change over time is also to say descent with modification. And descent with modification indicates relatedness among organisms. This diagram in figure 1.6 shows that when Darwin was thinking, he was thinking a way of how he could explain how all organisms were related to one another. And these branches show the beginning of this idea of the phylogenetic tree, the tree of life. When we talk about evolution, we're talking about changes in characteristics of a population over time. And these characteristics can keep on mounting up until you can have speciation. So species are related to one another and they can change through time. So what is a population? A population is a group of individuals of the same species living in the same place at the same time. Natural selection is the vehicle for change, and this explains how evolution occurs. So there must be two conditions for natural selection to occur. One is there should be variation, and this variation should be heritable. That means that this variation can be passed off from one offspring to another. And this also means in step 2 that in a particular environment, certain versions of a heritable trait can help individuals reproduce more than other versions. Certain heritable traits lead to increased success in producing offspring, and that causes traits to become more common in a population over time. So natural selection acts on individuals, and we have to remember that evolutionary change occurs at the population level. And again, when we have so many changes, speciations can occur. That means that there are two species, at least two species from one ancestral population. When we talk about adaptation and heritable traits, we always talk about fitness too. So fitness is the ability of an individual to produce offspring. Individuals with high fitness produce many more surviving offspring than do others in a population. For example, I think that I might be one of the most fittest people in the classroom. Now, I don't have a six-pack, but the truth is I have three children, and all three of my children are adults and they're able to reproduce themselves. So if you have three or more children, you are just as fit as I am. Okay, now why is it that I'm so fit enough to have three children? And it could be is I must have some traits that increase my adaptation and therefore increase my fitness to a particular environment. Another example of natural selection are finches on the Galapagos Islands. On some of these islands there's a high rainfall, and because of the high rainfall there's an abundance of small soft seed. And to pick at those seed and to get them, if finches have small pointy beaks, then they'll have higher fitness. In other words, the ability to survive and reproduce. Having small pointed beaks increases their adaptation and their ability to thrive on an island that has high rainfall. Finches with small pointy beaks increase in the population. So this is an example of an increase in fitness. When we talk about the tree of life, we're talking about relationships, genealogical relationships. In other words, when we talk about the tree of life or phylogeny, the evolutionary history of a group of organisms or species, we're actually talking about genealogy on steroids, because we're not just going back hundreds of years, we're going back thousands or even millions of years. So how is it that we can study this? We can study this by actually looking at the genetic variation. In other words, the variation of A, T, C, and G in specific sequences. If the sequences are very similar, then the species are related. If the sequences have a lot of differences, then you can imagine the species will be distantly related. So here's an example of comparing land plants to green and brown algae. So let's look at land plants to green algae. If you look at land plants and green algae and you look at the sequence ATATCGAG, you see that land plants are very similar to green algae, except instead of one C, there's a G. But if you compare land plants to brown algae, you see that there are many more changes than just one. Therefore, we can conclude that green algae is more closely related to land plants than it is to brown algae. The phylogenetic tree is used to show the relationship between species. If the branches share a recent common ancestor, then we know that species are closely related. If the branches do not share a recent common ancestor, then we know that the species are more distantly related. So the tree of life is based on and is estimated from genetic data and other types of data too. If we take a look at all life, we see that there's three clusters here, and these three clusters are represent three different domains: domain bacteria, domain archaea, and domain eukarya. And these clusters show that within the clusters, they're closely related. Outside of the clusters, they're more distantly related. Now, there's two types of cells when we look at the tree of life. There's eukaryotes and prokaryotes. Eukaryotes are found in domain eukarya. Eukaryotes have a nucleus. Eu means true and kary means kernel. Eukaryotes, true kernel. Prokaryotes are found in domains bacteria and archaea. Pro means before and kary means kernel. Before there was a nucleus. So those are prokaryotes. So both eukaryotes and prokaryotes differ in structure. In this figure, you can see a eukaryote cell at the top and a prokaryote cell down below. Notice the eukaryote has a nucleus while the prokaryote does not. Another way that we can look at life is through taxonomy, is putting organisms more than just into prokaryotes and eukaryotes, but into different taxonomic groups. And there's different levels of these taxonomic groups called taxons. So Carl Woese actually created or synthesized these idea of three domains: bacteria, archaea, and eukarya. But within each of the domains, there's kingdoms and phylums. Now, in 1735, Carl von Linné Latinized his name to Carolus Linnaeus, and he also gave organisms a Latinized name too that had two parts, and we call this binomial nomenclature. And the two-part system consists of a genus and a species. The genus is made up of closely related groups of species, and the species are individuals that can breed together within a group, or individuals that characteristics are very similar and unique and different from other species. So the scientific name or Latin name has genus and species, and it is italicized, and the genus is always capitalized. So a classic example would be Homo sapien. Homo, it's capitalized, H is capitalized, sapien is not, and Homo sapien should either be underlined or italicized. The last part of this chapter talks about the business of doing biology. The nature of science means that all scientists need to ask questions that can be answered by measuring things or by collecting data. In other words, having evidence of proof. So to start the scientific method, we need to formulate a hypothesis. Hypothesis is an educated guess that is testable and can be proven falsifiable. It's finding evidence that will either support or reject the hypothesis. Hypothesis testing has two steps. The first step is to state the hypothesis so that we can make a prediction. The second step is to design observational or experimental study so that we can collect data. And this evidence will be used to either accept or reject our hypothesis. So there's two examples in your book. There's two studies. The first one is, why is it that giraffes have long necks? So one of the reasons could be because of food competition. If a giraffe has a long neck, then it can have food resources that others can't reach up into high trees. So if we take a look, neck length is variable among giraffes, and this trait is heritable. So maybe it is likely that giraffes feed high up into trees. But the truth is data shows that maybe the food competition hypothesis is not supported by any data. So the third prediction that giraffes eat high up in trees may not hold true. If we look at figure 1.9, It shows that giraffes typically don't eat at very high levels. So we have two graphs here, one for males and one for females. And the x-axis shows the feeding height in meters, and the y-axis shows the percentage of feeding bites. And if you take a look at both of the graphs, the peaks of each of those curves are way below the average height of both males or females. So in the picture off to the right, you see that many giraffes are eating at levels lower than the height of their neck. And so maybe that's not a good reason why giraffes have such a long neck. An alternative hypothesis could be that giraffes have evolved long necks because long-necked males win more fights than shorter-necked males. Longer-necked males can then father more offspring, and for this reason, giraffes have longer and longer necks. And data does support this hypothesis. When you think about it, I think about working with my husband at home to fix things up. He usually has a really large hammer, and when he thrusts the hammer to hammer in a nail, he usually takes maybe 2 hits of the hammer and the nail is actually driven into a piece of wood. I have a smaller hammer. I'm going to be honest with you, I have to hit the nail on the head several times before I can drive the nail into the wood. So the longer the handle, the more force can be used to hammer in a nail. So we can see that longer necked giraffes have a fighting advantage over those that have shorter necks. Another experiment is, how do ants navigate? So there's an idea that if ants leave their nest, how do they know how to find their way back home? How is it that they can find their way back home? One idea is that they can count the number of steps that they take, and that's how they know how to get back home. So this is called the pedometer hypothesis, where you take into account the stride of the ant and the number of steps that it takes. So both stride and the number of steps leads to the pedometer hypothesis. The null hypothesis means that that ants are not using or counting steps to find their way back home. So null means the opposite of what we think. Steps has no effect on getting way back home. So here in figure 1.10, you can see how is it that an ant finds its way back to the nest after foraging for food. Are they counting their steps? So in this experiment, there's 3 treatments and everything else is controlled. In this case, the treatments affect the stride of each of the ants. So there's stumpy legs, normal legs, and stilty legs. For stumpy legs, the legs were cut. For normal legs, they were left alone. And for stilty legs, bristles were glued onto the legs. And we wanted to know if the stride affected their navigation skills of getting back to the nest. When you take a look at the data down below in the graph, you see on the left-hand side that at 10, that is getting back home. So when you have normal legs, that's indicated in green, ants will find their way back home. But if they have stilty legs, they'll overshoot, or if they have short legs, they'll be short and they won't be able to make it home. So here you can see stumpy, normal, and stilty legs on ants, and it does affect their ability to find their way back home. The null hypothesis specifies what we should not observe if hypothesis being tested does not hold. In this case, stride number and length did have something to do with navigation, but the null hypothesis states that it will have nothing to do with navigation. So in this case, we can reject our null hypothesis. Conclusion: desert ants use information on stride length and number to calculate how far they are from the nest. So the data supports the pedometer hypothesis. Other characteristics of a good experimental design include a control group. So the control group is often the no treatment or the normal group. In this case, with the ant experiment, normal meant legs that weren't tampered, not too short and not too long. Experimental conditions must also be constant. The only thing that's changing is the treatment. And all good experiments have repetitions. So repeating a test is essential, and also having a large sample size. This is the end of Chapter 1.